Let me begin by thanking the organizers too for the invitation to speak here. I think it'll be an interesting workshop, and I'm looking forward to some stimulating discussion. So I'm going to provide an introduction to quantum mechanics of photosynthetic light harvesting. You can't quite see my title, but it's quantum mechanics of photosynthetic light harvesting machinery. So I'm going to provide a bit of introduction and then talk about some of the work in our group in this area. Okay. So, right. And since this is a quantum biology workshop, I thought I'd motivate a little bit and give, start at the beginning and talk about quantum biology and what it should mean or what it does mean. And the way I like to think about it, I like to classify it into three levels of quantum influence in biological systems. So going down the list here, I mean, the first one is the trivial. Since we believe quantum mechanics is the theory of microscopic systems, then everything is quantum mechanical at some level, right? But that's at this level of reductionism, you can't really add much to knowledge. Quantum mechanics dictates energy levels, molecular orbitals, and so on. But does that really tell us anything new? The next level up, which is more interesting, is the level of molecular dynamics and chemical kinetics. So, for the vast majority of systems of biological interest, you can describe chemical kinetics, molecular dynamics by just classical physics, right? But in a few cases, there are quantum corrections which turn out to be significant. Just some examples here are these non-adiabatic transitions through conical intersections, which are a popular area of research these days. I mean, you need to have quantum mechanics in order to describe the rates and the correct transfer in these cases. And then there are chemical reactions involving tunneling of electrons and maybe protons. There again, the quantum corrections become important, right? And then the next level, which is maybe the most tantalizing, is where quantum mechanics becomes a functional necessity. Okay, and the division between these two is kind of hazy. Where you throw one and where you throw the other is not clear. But at least the third class of quantum influence, you should be able to make a connection between if the quantum mechanics wasn't there, if the system was entirely classical, the organism wouldn't be able to survive, right? And I've listed some examples, or some potential examples from this class. The way geckos hold on to things is maybe through van der Waals forces, magnetoreception in birds, olfaction, and finally photosynthetic light harvesting, which is what I'm going to focus on. I should say that the last class is really the only one where we have convincing experimental evidence for quantum effects. You could still debate over whether it belongs in class 3 or class 2. I think I'll talk more about the role of quantum mechanics in light harvesting, but I tend to put it in class 3. Okay. So then I should also go over the skeptic's view, right? This is if you say to even the normal scientist, you expect quantum mechanics to play a role in biology. There are a few arguments for why this should not be the case that most people bring up. The first of these is a classical kind of thermal argument. They say that there's too much thermal energy in biological systems, right? The typical energy scales that you're concerned with are just overwhelmed and drowned out by just the thermal noise and thermal energy in a biological system. Okay, and that's a fairly weak argument in my point of view, because it's a very equilibrium argument. I mean, the temperature is an equilibrium concept, right? And in most biological systems, it's the non-equilibrium dynamics that matters. And you can have systems that take a very long time to actually equilibrate, and yet you can be very quantum mechanical in that region before equilibration or before thermalization, and still have a state that's thermal at the end. Okay, so this is not a convincing argument, I don't think. A more sophisticated way of putting this comes in the framework of decoherence theory, where you really consider the timescales in the system. You say there are two timescales that matter. One is the timescales that of interactions within the system. And by system, I mean the biological thing that we're looking at, the structure that we're looking at, and that's of interest, right? That's parameterized by this J. So this is the rate at which dynamics occurs within the system, and 1 on J is the coherent evolution timescale. And then there's a rate of interaction with the environment, and that's parameterized by this lambda here. And so you can see that what J does is it likes to create superpositions between states, between classically distinguishable states. Okay? What lambda does, it likes to decohere these superpositions into classical mixtures. Right? And so there's an intrinsic competition between these two rates. And so a nice way to talk about how quantum the system is to think about this ratio, J on lambda. Okay? And so you can draw this scale down and say that, well, we know that all the way on the right, where J on lambda is large, is where things are very quantum mechanical. And here we have classic systems from well, these days from quantum computing, trapped ions, superconducting systems, maybe photons and atoms in optical lattices, and so on. Here, J on lambda can reach tens of hundreds. On the other end of the scale, and this is typically the skeptic's view, right? That this is where biochemical processes lie. They lie all the way on very small J on lambda. Okay. But one should ask the question: is that a priori necessary? I mean, should biochemical systems only lie on that side? And what I will hope to show you is that we know of at least one biochemical system which is much closer to the right-hand side of the scale. Okay, and maybe there are more. Okay, so that brings me to the main topic of the talk: quantum effects in lighthousing systems. I'm going to provide a little bit of introduction in the absence of Alain's talk here, so bear with me if you already know this stuff. So the structure of photosynthesis really is, you can break it down into two sections, what are typically called light reactions and dark reactions. The dark reactions are the chemistry, essentially. The Calvin cycle, everything that goes from sugars, everything that goes from units of biochemical energy to sugars. And then the light reactions, which is what I'm going to focus on, is what the things that are initiated by light, by photons. Okay? And if I zoom in on those light reactions, this is a cartoon picture of what happens. You get light impinging on absorbing pigments. There could be a whole variety of them going over multiple energy scales. So we've got blue, orange, red. Eventually, the resulting excitation migrates its way to something called the reaction center, which is where most of the chemistry is initiated. Okay, so this is where you get a setting up of an electrochemical potential, which is essentially a battery for the rest of the reactions. So I'm going to now zoom in a little bit more and just focus on this first part, what are called the antenna systems. These are the things responsible for absorbing the light and also eventually transporting the resulting excitation to the reaction centers. Okay, so there's a huge variety of antenna centers. This is just a picture from Blankenship's book. If you want to make generalizations, then some things you can say is that they're all composed of densely packed pigments. So pigments are things like chlorophyll molecules, which absorb light very efficiently. Okay, and most of these pigment or molecular aggregates, or these pigment aggregates, are encased in these protein cages, such as these that you see, which lends some stability and some energetic structure to the system. Okay. And all light-harvesting complexes are also embedded within membranes. And I'll come back to this point at the end, why this could be important. Okay? So another generalization you can make, which is perhaps the most important at this point, is that energy transfer is very efficient in these systems. Greater than 95% efficient in most cases. And it happens at very fast timescales, picosecond timescales. Okay? So these are some of the fastest processes we know in biology, and arguably some of the most efficient as well. So let me introduce you to a specific light harvesting complex here. I'm going to concentrate most of the theoretical studies later on this complex, so I'm going to go through in a little bit more detail. This is the apparatus of green sulfur bacteria. So these are bacteria. These are anoxygenic, so they don't produce oxygen, but they live in these sulfur-rich environments, typically in oceans or swamps. This is a picture of them. If you zoom into the light harvesting apparatus, it looks like this. It's essentially, most of the light harvesting, the antenna of the system, is this large complex you see at the top here. This is called the chlorosome. It's composed of very densely packed pigments. and that's where most of the light is absorbed. And eventually the resulting excitation from the absorption migrates its way down to the reaction center, which you can see here. Can people see the cursor on the screen? Yeah. Is that okay? Okay, all right. I'll keep using that. So in this migration of this excitation energy, right in the middle is this thing called the FMO complex. Okay, so it's essentially a wire. That channels the energy from the main antenna to the reaction center. Okay, and I'm going to zoom in on that a little bit. And has this structure. It's a trimer of identical units that looks like this. If I just look at one of the monomers, this is its picture, and if I strip away the protein cage, all this gray matter around it, it's essentially just 7 chromophores, 7 chlorophyll molecules. Okay, there's a standard labeling 1 to 7, and the way I've drawn it here is actually what we believe it looks like in vivo. So this is the orientation. The light enters through chromophores 1 and 6 from the base plate, and it leaves through chromophore 3. Right? So it really is just a wire that connects the main antenna to the reaction center. Okay, and it is very efficient. It's amazingly efficient at this job it's been given of funneling this energy down. So as you probably all know, there were experiments on this, precisely on this complex, on the FMO complex in 2007, which showed that not only are the static features quantum mechanical, but the dynamic features in the system are also quantum mechanical, meaning that how energy moves about among these many different pigments is not by just hopping from pigment to pigment. It rather moves around in a wave-like manner. It samples a lot or many of the pigments at once, okay, as it's moving around. And if you look at, so I mean, I like to think of this as basically getting, refuting this classic picture of energy transfer in these systems, of a excitation jumping from pigment to pigment, and rather moving to this other picture of where this is now an energy space. These are the energy levels of the system, and it's just coherently sampling the energy levels. It's moving up and down more like a wave. Okay, and that's a very cartoony picture, but I think it's useful to think about the sea change in the picture in which the dynamics is happening. Okay. Yeah, and then as Hartmut mentioned, there have been several experiments since then. So this initial experiment was done at cryogenic temperatures, and now there's been multiple experiments at room temperature which confirm the same effect, and in multiple organisms too. So not just in green sulfur bacteria. This is not an outlier in the space of lighthousing complexes. Okay, it's been confirmed in marine algae, in purple bacteria, also in higher plants. So LHC2 is a lighthousing complex present in spinach. Right? So in most of the green plants that we see contain LHC2. And we know that there are quantum coherent effects in LHC2. Okay, so now moving on to the theory, there's been a lot more theory on these systems, and this is just a small sampling of the theoretical papers out there. People have looked at efficiency of these systems and how quantum mechanics plays into the efficiency. I think Masoud will talk a little bit more about this in the next talk. People have also looked at more exotic quantum properties like quantum entanglement and speed up in a dynamical sense. And I'm going to talk more about these two topics later on in the talk. But just to survey, there are other things that people have looked at, and Masoud will again present some more on this, is optimality of the system. Is it really tuned to do what it's meant to do, or is it not really optimal at all? And I'll let Masoud answer that question, because since he's done the work there. And then on purple bacteria, which are in a sense more heavily studied, because I guess the quantum mechanical effects there, at least the static quantum mechanical effects, were known for a longer period of time. People have looked at delocalization, so how coherently delocalized is the energy in the system, how the transfer rates between modules of this purple bacterial lighthousing complex dictated by quantum mechanics. And also in higher plants, people have looked at entanglement in these lighthousing LHC2 complexes of higher plants. Okay, so now I'm going to move on to some of the work that we've done specifically. And I'm going to begin with looking at entanglement in the FMO complex itself. So let me define entanglement, give a brief introduction. So entanglement mathematically is this statement that if I'm going to write down a quantum state, if I can write it, and it's a quantum state of many parties, so I've divided my system into a bunch of subsystems, and there's some natural division. If I can write it down, if I write down my quantum state as a state of party A, tensor producted with a state of party B, tensor producted with a state of party C. If you're not familiar with the tensor product, I mean, it's just a mathematical term for saying this independently of this, independently of this. So if you can write down the state of the combined system independently of, in a way that each system has its own independent state, then we call that a separable state. And then any state that's not written this way is an entangled state. Okay? And so I've written an example here of an entangled state. So this is, these are spins, spinning pointing up or down. So you can see that you can't factor out the up component of the spin state, right? If you factor it out of this state, you can't factor it out of here. So this is party, the left hand side is party A and the right hand side is party B. Okay, so that's curious. You might think that's nice math, but why does it matter? And really, to understand the fundamental importance of quantum mechanics, you have to think about what it means for what the state is telling us about knowledge. Okay, I think this is the clearest way to interpret the system. So the entropy of a state, we think of how much uncertainty there about the state of the system there is. So it's a measure of uncertainty. Just like classical entropy is a measure of uncertainty, you can define a quantum entropy, the von Neumann entropy, it's a measure of uncertainty. So that's this entropy function here, there's S of rho. If I calculate the entropy of this entangled state here, it's 0. Okay? So in a sense, I know as much as I can know about this state. Okay? It's a pure state, and I know as much as I can know about this state. But now if I ask the question, what is the state of either party, of party A or party B, the natural way to think about that is to average over the state of the other party. Okay? It's called the partial trace operation. And this is what you get for the state of either individual party. It's a completely mixed state. It's a classical mixture of up and down. And if you look at the entropy of that, it's 1, which is the maximal entropy such a system can take. So what is this saying? And this was first noticed by Schrödinger way back in 1935. And he says what this means is that the best possible knowledge of the whole does not imply the best possible knowledge of its parts. Okay? And this is the reason people get excited about entanglement, or people are perplexed by it, because this is very counterintuitive. If I tell you I know everything about everything in this room, I should be able to answer the question about what state is that chair, what color is this chair, right? But that's not the case for entanglement. Even if I know everything about the global state, I can have as little knowledge as possible about individual states. Okay, and so this was recognized a long time ago, and this is precisely what led Einstein, Podolsky, and Rosen to maybe posit that the quantum mechanics is not a complete description of reality. We've progressed a lot since then, and we think that it's a complete description, but maybe one that's not intuitive. Okay, and then more recent developments, some manifestations of entanglement. These days, entanglement is not. It's still weird, but it's not necessarily something we don't get our hands wet with. Experimentalists create this all the time. There are experiments with superconducting systems, ion traps, photons that routinely create entangled states. And furthermore, we know that it's useful for certain tasks: information processing, metrology, communication, and so on. But I should note that these are all low-temperature, low-noise experiments. To create an entangled state with a high fidelity requires a lot of effort from experimentalists. And even small deviations from ideal conditions can destroy entanglement. So, but there's a more recent developments that show that even bulk systems, if I just take a magnetic salt in this case, if I talk about it's the state of its constituents, the nuclei of this magnetic salt, below a certain temperature, I think in this case it's about 1 Kelvin or 5 Kelvin, we know that the state of the nuclei are entangled. Okay, we can do bulk measurements which tell us that the most consistent state is an entangled state. So in a sense, this is a natural system. It's just a magnetic salt that is entangled below a certain temperature. Okay. so it kind of led us to ask the question, can it exist in biological systems, which is in a sense a more natural system, and at physiological temperatures, right? So this was below a certain temperature. Can we see it in biological systems at physiological temperatures? And that's the question I want to answer today. Before I do that, let me clarify what I mean by entanglement in these light harvesting complexes. So I've shown you that they're mostly composed of chlorophyll or pigment molecules that are spatially separated. And by entanglement, what I mean is that non-classical correlations between the electronic states of spatially separated chromophores. Okay? So chromophores are spatially separated, and they have some electronic degrees of freedom which are moving around. And if you have non-classical correlations between them, that's what I'm going to call entanglement. This is a fairly sensible definition of the concept. The only thing is how you divide up your whole system. And here I've chosen to divide up the degrees of freedom are the degrees of freedom of the chromophores themselves. And this is sensible because they're actually spatially separated individual components. So how do you quantify entanglement in such a system? And it turns out to be very difficult. Quantifying entanglement is a tricky concept by itself. And in this case, what we have is something called multipartite mixed-state entanglement. I'll explain what those things mean. Multipartite, just because you don't have just two parties here, right? You have many chromophores. You have at least 7 in this FMO complex. So you can't just talk about a bipartite system. And it's mixed-state entanglement because the system is never in a pure quantum mechanical state, meaning that it's always interacting with its environment. It's a biochemical system, after all. It's interacting with its environment, and the state of the system is never in a pure quantum mechanical state. So we have to talk about mixed state, multipartite entanglement, and there is a simplification which we can make, which makes us able. It allows us to actually quantify the entanglement in the system, and that's that. We're always in the single excitation subspace in the system. What I mean by that is that this, particularly for this FMO complex, and more generally for many light harvesting complexes, the amount of photons that they receive per unit of time is very small. So you can be very sure to a very high level approximation, you can say that there is at most a single excitation in the system at once, right? It transports excitations in femtoseconds and picosecond timescales, and it receives a photon once every seconds. So it's gonna dump that photon much before it gets a long time before it gets the next one. So we're going to use this approximation that there's at most a single excitation in the system, and what that and that's very accurate for at least this FMO system, and more generally for other light harvesting complexes as well. And that allows us to form measures of entanglement in this system. I'm going to use 2 measures. One is a global measure of entanglement, so it kind of captures the total amount of entanglement in the system. Okay. I'm not going to go through the technical details of how you come up with these measures or how to interpret them, but I'm happy to talk about that at the end if there's time. And then the other measure of entanglement I'm going to use is a standard measure called the concurrence. It's just a measure of bipartite entanglement. So I single out 2 particular chromophores and I ask how entangled are they. Okay, and I'm going to use a measure of concurrence for that purpose. And okay, so now I've talked about how to quantify entanglement. I'm going to talk about how to actually theoretically simulate these systems so that you have some way of actually calculating how much entanglement there is. And this is maybe too technical for people who aren't interested in the details, but I'm just going to go through it very quickly for people who are. A very good approximation of the dynamics here is what's called the Frenkel Hamiltonian. What it says is that each chromophore, so this notation here of n, you should think of that as the state of a single chromophore, okay, the state of a single pigment. What it says is that each pigment has a certain characteristic energy to it. This is the energy of its excited state, and there's a matrix element that couples pigments, and this is a physical Coulombic coupling. So if there's an excitation on here and there's a nearby chromophore right next to it, it will eventually move over to here. Okay, and and how quickly it moves over to here is characterized by the J. Okay, so it's technically called the transition dipole couplings. So that describes the movement of excitation in these systems, but that's not all, right? Because I've mentioned a couple of times that it's actually embedded within this protein cage. It's embedded within a lot of other dynamics happening around it. So you need to actually talk about the open system dynamics, and that's the Hamiltonian there. What describes the dynamics there is not only this part, but also something that describes the coupling between the chromophores to the environment, and also the dynamics of the environment. that ends up being a lot of things to keep track of. So what the usual way to do this is to average out over the dynamics of the environment. What you say is that I'm going to ignore what the environment is doing, and I'm just going to look at the system of interest. And that's you compose things like the master equation, which describe the dynamics of the system. Now, the tricky thing about this particular system and about lighthousing complexes in general, and we could talk about why this is that there are no perturbative timescales in this system. The standard way of going from this complete description of the system and the environment, everything around it, and just focusing on the environment is to perturb around some timescale. You say that the environment is not moving very quickly, so I'm just going to say it's a constant. It's constant or moving very slowly, right? And this is a standard mathematical technique. You separate timescales, and you just talk about the timescale of interest. You ignore things that are happening too fast, and you ignore things that are happening too slow. But the thing about these systems is there are no perturbative timescales. It's really difficult to do this. And so you need a theory that can take this into account. And very recently, just last year, such a theory was developed in the Fleming lab at Berkeley, and Akihiro Izusaki was the first author there. And we're going to use that model for the dynamics. So we're sure that we're capturing the dynamics of the system in the regimes that are interesting, precisely in this regime where there are no perturbative timescales. Okay. All right, so here are the results. So these come out of running the simulations according to this very accurate physical model, and measuring the entanglement using the measures that I mentioned. So if we just look at global entanglement, right? So this is the total amount of entanglement in the system. The two curves here, the blue curve is for 77K, which is where the first experiments were done, and the red curve is for the room temperature, 300 Kelvin. What we see is that there's an initial spike of entanglement. So the initial state here is that the excitation is just on one of the chromophores. So here I mentioned that the FMO receives its excitation through 1 and 6, right? So I'm just choosing the excitation to be on 1 in this case. And then as it's spreading out over the chromophores, this entanglement build up over all the chromophores that are involved in the transport. And you see that the temperature dependence is not drastic. So there's a viewpoint that increasing temperature will kill off quantum effects very quickly, right? But this is not what you see here. You've increased temperature almost fourfold from 77K to 300K, and the entanglement has decreased by maybe 3/4, right? So there's no kind of linear or superlinear scaling of I should say there's no superlinear scaling of the suppression of quantum effects in this case with temperature. And furthermore, there's significant entanglement at long times. It's at the steady state of the system, which is way over here. So sorry. So the main graph shows time up to 1,000 femtoseconds, and this subgraph is showing times up to 5,000 femtoseconds. So there's entanglement even up to 5 picoseconds. Now that's on absolute timescales, or at least on human timescales, that's very short. But you have to remember that these systems these things do their job in femtosecond timescales, right? So these are certainly functionally relevant timescales, and that's the important thing to keep in mind. Okay, so as I mentioned, this is a measure of just the total entanglement in the system. It doesn't really tell us about what things are entangled. You might say, well, this is maybe not so surprising because you have chromophores that are very close to each other. They're talking, they're strongly coupled to each other, and therefore they will have entanglement with each other. So maybe this is not so surprising. And to look at more closely at where exactly the entanglement is spatially, what you have to do is resort to this kind of bipart type measure. So I'm going to choose 2 chromophores and then just look at how entangled they are. Okay? And so let's just focus on the left-hand graph here, the 77K. I think you can see this. This is showing which 2 chromophores each line corresponds to. So the blue is chromophores 1 and 2. The red is chromophores 1 and 3, and so on. And so you see that the intuition was right there. The chromophores that get very entangled quickly, and there's a lot of entanglement, are the ones that are right next to each other, 1 and 2. Okay? But then, and then as the excitation moves around, other things get entangled. And the really surprising thing for us here was this 1 and 3, the red curve here. You see this entanglement build up over the chromophores 1 and 3, and it stays around for long time scales, right? At least until a picosecond at 77K and further. So that's this red curve. And why this is surprising is that 1 and 3 are almost as far apart as they could be in this complex. They're about 3 nanometers separated, and they have negligible direct coupling. Right? If you just look at the direct coupling between these things, they're not very coupled at all. So this entanglement, this non-local correlation between these two, is mediated by the interaction with the rest of the system. Okay, and so and if you look at I've just plotted the curves which show the most significant amount of entanglement. But if you look at other bipartite cuts as well, they also show entanglement. And what it means is that there's true multipartite entanglement. So if you look at entanglement theory, people make the distinction between things that are just bipartite entangled, and for 3 systems, only these 2 are entangled, or are they all entangled with each other, which is a more complex description of the entanglement in the system. And it turns out that it's multipartite entanglement in this case. Okay, so that brings me to the end of the entanglement discussion. Now I'm going to talk about quantum speedup. So what do I mean by quantum speedup? We were motivated to look at this by statements like this that came out right after the experiments were done in 2007. So people made the analogy to quantum computation in these systems, saying that, well, planets are actually performing a quantum computation in the process of absorbing energy. And for anyone who works in quantum computing, that's my background, this is a very striking statement. If you're talking to experimentalists, they've spent years and years, building up a single qubit to do virtually nothing. And so, it's very kind of almost insulting if you talk to a quantum computing experimentalist to say that such a messy system is performing a quantum computation. So we wanted to really examine this question closely. And the way we do it is, so the most direct analogy to a quantum computation is through this thing called a quantum walk. So quantum walk, most of you probably know about a random walk. A random walk is just where you move randomly between spots, and then you eventually diffuse out in a system, right? So if I have a line, and I move to the right with probability p, and I move to the left with probability p, after a certain amount of time, I'll have a Gaussian distribution for my position, right? This is classical diffusion. If now instead I say that I'm a quantum walker, I can move in superposition to the right and left. If I look at my distribution after a certain amount of time, it'll be this black curve. So the blue curve is the classical distribution, it's just Gaussian. It'll be this black curve, and it's really strange. Not only is it oscillatory, it's also bimodal, right? So you have maximum probability of being on the ends, not in the middle anymore. So if you just look at the variance of that walker, it scales linearly in time, as opposed to the variance scales linearly in time, right? Or quadratically. Yeah, the variance scale quadratically in time, I think. But it scales polynomially faster than the classical walker. Okay, the variance of the system. And you can embed a quantum algorithm, like a quantum search, within a quantum walk. So you can say that the walker is searching for a particular node in the graph, and you can embed the whole idea of Grover's search algorithm within a quantum walk. And this is the most natural way in which to think about photosynthetic systems as performing a quantum computation. They're actually randomly walking around this chromophoric complex, and maybe they're doing some computation in this process, right? But one thing to keep in mind is that this is not an ideal quantum walk at all. For an ideal quantum walk, you assume that all the nodes are at the same energy. There's no decoherence. And in these systems, there's a certain energy landscape. And you need an energy landscape because you need energy to be funneled to reaction centers, right? So there's a certain energy landscape in the system. There's disorder in the system. There's also decoherence in the system, which we've talked about extensively. So can you really have speed up in this environment? Can you really have this kind of quadratic speed up over propagation in this environment? And to look at this, what we did was actually look at the FMO complex and map it to a 1D quantum walk. Since we know the properties of the 1D quantum walk very well, if you look at the Hamiltonian, so what does this Hamiltonian mean? The diagonal elements of the Hamiltonian are the energies of each chromophore, and the off-diagonal elements are how strongly they're coupled. Okay, so this element here will tell you how strongly chromophore 1 is coupled to chromophore 2. Okay, now if you just get rid of the smallest elements and you just look at the largest elements here, what you notice is that it looks very much like a 1D quantum walk. You have 2 source nodes, 1 and 6 at the ends, and you have a sink node in the middle, 3. Okay? There's a slight ambiguity here that there's 5 and 7 are equally or very strongly coupled to 6 and also 4, but you can collapse those 2 into a single node in a quantum walk. And you have a 6-node quantum walk, and you have things coming from the ends and moving to the middle. Okay? And now, a good way to see if you have a quantum speedup is just to say, look at the scaling of the variance, and say if it's classical, it's going to scale linearly, and if it's quantum, it's going to scale quadratically, right? So it's direct analogy to this picture here. Right? And so what do you see? So So on the left-hand side is a graph of the actual the variance with respect to time, and the right-hand side is its power law. I think the right-hand side is probably more, it's easier to interpret, but we can look at both. Let's see. So in the what you see is that up to about 70 femtoseconds, you have ballistic motion. So you truly have a quadratic scaling of variance. It moves very quickly, right? So it's easy to see in the power law. The power, if you just look at t to the b is 2, right? So you have quadratic scaling of how quickly it's moving. But then very quickly, it goes down. It goes to actually sub-diffusive. It goes to below what you expect for a classical random walk. And then it eventually gets trapped in the trapping sense. So this includes trapping. That's why it's going down continuously. But what this tells you is that there is no quantum speedup in the sense that you need for a quantum computation beyond 70 femtoseconds. Okay? But the coherence is still there up to 700 femtoseconds, an order of magnitude greater. And you could ask, so why is this happening, right? What is limiting the quantum speedup? And it turns out it's a combination of two things. One is it's localization. And this is well known from condensed matter, and it's usually called Anderson localization in very large systems. It's where if you have any disorder in your system, so if it's not a plane, if you're not just moving along an energetic plane, if you have like disorder in your system, you're going to get localized. You're going to get trapped in local minima. You're not going to propagate very far in the system. But at the same time, there's a dephasing mechanism, which is the effect of the environment. And what the dephasing does is it moves energy levels around. Okay? So if you imagine a static picture where the energy levels are just fixed, you're going to get trapped. But then the dephasing actually helps you because it moves energy levels around, and you can get pushed over boundaries. And so the dephasing moves you beyond being trapped. So if you didn't have the dephasing, this would go from 2 and rapidly drop down to 0. Right? So the effect of the dephasing is to push it back up, but it still doesn't push it back up to 1. So it's actually sub-ballistic all the way through after 70 femtoseconds. So we think this is a pretty convincing argument for there is no quantum speedup in this system. The combined effects of the disorder, the localization, and the dephasing preclude any kind of quantum mechanical speedup that's required for quantum computing in this system. Okay? Okay, so how much time do I have left? 15, great. Okay, so I'm going to talk a little bit more about. Maybe this question is naturally motivated at this point. What's so special about lighthousing complexes, right? I've presented you some results, and you'll see some more about their efficiency and so on. But I want to address what is special about lighthousing. These are the only, arguably, the only biological systems where we know quantum effects play such an important role. And so what makes these things so special? And this is not just a philosophical question. I think this is a very practical question, because if we want to find more systems in biology which might benefit from quantum mechanical effects, then we should really isolate what is special about these systems. We should really find out what makes these different from any other biological system. And so it's useful to go back to this picture that I introduced about the system and environment coupling. So J is the rate at which dynamics happens within the system, and lambda is the rate of dynamics between the system and the environment. And it turns out for light harvesting systems, they increase J by doing a very nifty thing of just having very dense packing of molecules, right? So these chlorophyll molecules are packed very close together on separation of lengths of a nanometer or sub-nanometer length scales. This leads to very strong interaction between them. So J is kind of pushed up naturally by this engineering in a way. And furthermore, lambda is pushed down. And there's a couple of tricks that it uses to do this. So the main trick is that the light harvesting complexes are always embedded within membranes. Okay, so you have some membrane, and the light harvesting complex lives within the membrane. And there's a very good reason for this. It's really to set up an electrochemical potential. If you want to separate electrons from protons, positive charges from negative charges, you need to find a way to keep them separated. Otherwise, they'll recombine again. So what you do is you send the positive charges to one side of the membrane and the negative charges to the other. Okay? So this is the membrane, in a sense, is a battery here. The terminals of the battery. But maybe it's doing something more. If you put things inside a membrane, they're in a more controlled environment than just in some bare solvent. Okay, and it turns out if you look at how strong the electron-photon coupling, how strongly the chromophores are coupled to the vibrations in the system, it's orders of magnitude smaller than what you'd expect in a typical solvent. So if you look at what you'd expect, this coupling is called the reorganization energy. If you look at what the reorganization energy that you calculate in a typical solvent, it's about 200 to 2,000 wave numbers in this unit. And if you look at, for the FMO, what it is, it's about 35 wave numbers. So it's drastically smaller than what you see in a typical solvent system. And we believe this has to do with the membrane embedding. It's a much more controlled environment. And another aspect that comes out of this is that maybe the fluctuations that it sees are much more controlled. They're not just random fluctuations. There's some correlation time, and there's some spatial correlation to the fluctuations as well. And people, including Masood, have shown that this can actually help in the transport. If you have correlated fluctuations as opposed to a completely random environment, you can preserve coherence for a longer period of time. Okay, so this could be What does this mean? It means that for these systems, J on lambda is about 1. They don't lie all the way on this left-hand side of this graph that I drew. Maybe this is an engineering choice, right? You push J up and you push lambda down in order to achieve this kind of coincidence of timescales. Okay, so this leads me to structure, and this is some work that we've recently started. I'm going to talk about some of the work that we're doing now and some of the things we're looking at. These considerations lead you to think about structure in these systems and how structure plays a role. And so in biology, I guess these are normally called structure function questions. You want to know, why the structure is so that it performs this function. And you can really kind of form this picture of lighthousing complexes going from a monomer or a signal pigment. You aggregate them, you form them together into a block, and then you put some protein cage around it in order to give it some stability. Okay, so how does each aspect of this construction affect the quantum mechanical effects and also the function of this device? So you can ask, which structural features actually determine the quantum behavior? What's the influence of the pigment density? What's the influence of the pigment orientations that you choose, of the actual choice of pigments that you use? You can ask whether you can engineer the protein backbone. So I mentioned that the membrane may be a way that you have some shielding from environmental effects. Can you engineer the protein backbone so you have this similar kind of shielding? And how can you enhance quantum behavior by suitable design? And so how does the aggregation, how does the protein determine both the transport properties and also the optical properties? So I haven't mentioned much about the optical properties, but really these things are antenna, right? So not only do they need to be good at transporting excitations, but they also need to be very good at absorbing the light that they receive. It's a whole other story. It turns out that most light-harvesting complexes are really finely tuned to the actual spectrum that they receive. So to investigate these questions, what we're doing is actually building test systems. We're basing this on a tobacco mosaic virus. So this is a common virus. This is just a monomer of the protein. And what you can do is you can mutate certain sites of this protein, and you can add chromophores. You can add pigments. Right? So the pigments will be attached to these sites, and then they self-assemble into these beautiful structures. So they self-assemble into these. You just throw a bunch. So you mutate a bunch of them. You attach chromophores, and then you throw them into a solution with a certain pH and temperature, and they self-assemble into either these disc-like structures, these stacks of discs, or they can even assemble into these helix structures. Okay, and these look very similar to natural light-harvesting systems that we have. But there's a natural light-harvesting analogy counterpart to these helical structures and also to these disc-like structures. And so the person doing the synthetic chemistry is Matt Francis at UC Berkeley. He's a professor in the chemistry department. And so he has worked with these TMV systems for a long time. He's really an expert in designing the structure and also assembling it in the way. And so what he can do is mutate different components and attach chromophores at different sites to achieve variable separation distance between chromophores. So what does this mean? If he attaches it at site 103, right, they're separated by only about 0.8 nanometers. And if you make a disk out of them, they'll lie right in the middle here. Okay? But if he attaches them at site 123, the green site, I guess the colors don't come out so well here, but the point is that where he attaches them determines what the density of pigments is and what their orientations is. Okay? See, we can kind of make light harvesting complexes on demand here. And that's the idea. So we call this a bottom-up approach. We're really trying to understand the structure-function relationships. Like if I make a lighthousing complex like so, how do the quantum properties emerge out of that, and how do they influence function, like optical and transport properties? Right. So the goals here are, I guess, to mimic the fantastic efficiency of natural lighthousing complexes, but by design, by really microscopic design, and also give it tunable properties. So one of the things about natural lighthousing systems, they don't use a huge variety of pigments, but by just engineering the pigments so they can absorb either all the way on the blue or all the way on the red. And so can we have that kind of variability? Can we tune the absorption of these lighthousing systems by just changing the structure, by engineering it like so, by not only changing the pigments? And also we want structures that are suitable to biological environments. And obviously there are applications to this, maybe light sensors, also photovoltaic applications. Okay. All right, so here's a summary of what I presented. So the first part I presented, the entanglement. Hopefully I demonstrated that entanglement is a natural feature of these systems. It turns up as a result of the excitation transport. What I didn't really address is the role of the entanglement. So we haven't been able to pinpoint a precise role for this entanglement. It could be just a side effect of the coherence or the fact that they're strongly coupled. So we're still working on that. And the class of entangled states, for people who know about entanglement, the class of entangled states that these things exhibit are a very small class of the total entanglement that they could exhibit. It's only the so-called single exciton manifold. And you could ask whether there's more general class of entanglement that they could exhibit as well. The second part, I demonstrated that there really is no quantum speedup in the quantum information sense in these systems. It's not, these things aren't built for quantum computation. And the effects, the natural effects that they have don't allow for the kind of quantum speedup that's required. But that's not to say that there's no quantum advantage. You should make a distinction here. The quantum computing is not the only role here. Right? As Hartmut mentioned, that really overcoming local energy minima is a possibility. People have postulated that efficiency is a role, robustness, coherence could provide more robustness in these systems, unidirectionality. So you want these things to take energy in one direction towards the reaction center. You don't want it to go backwards, right? So maybe that's a role for the coherence as well. And then I've briefly presented the new approach that we're taking, the bottom-up approach of actually constructing artificial lighthousing systems and playing around with them to see what the effects of the coherence are. Okay, thank you. And let me... All right. This was, so this work was done in collaboration with a lot of people. The speed-up work was done in collaboration with Stefan Hoyer. He's actually here today. You can talk to him about if you're interested. The entanglement work was done in collaboration with Akihiro Ishizaki and Graham Fleming. And the synthesis work that I talked about at the end, the molecular synthesis is done in collaboration with Dan Finley and Matt Francis. And Brigitte Whaley was involved in all the work as well. Okay, thank you.
You had the entanglement scaling it with the power law with exponent 3/4, right?
The speed up, the variance.
Yeah.
The variance.
I'm interested in how does that exponent itself scale with the difference between the energies that you're dealing with, which is several electron volts, and KT at room temperature. I guess what I'm saying is, if you suppose that photosynthesis happened at 2,000 wave numbers instead of whatever it is, 15,000, would a different exponent apply?
So this is actually transport within the smaller wave. So this is transport within the excited state manifold only, right? So the ground to excited is about 15,000 wave numbers, but within the excited state manifold, it's only about 1,000 wave numbers or so separation. So it's the transport within that gradation. If the energy difference was even larger, it would be even more localization. You wouldn't have dephasing mechanisms which could bridge the gap of 15,000 wave numbers. I think that's.
I guess what I'm saying is that one of the reasons why the photosynthetic apparatus has been the first place where these kind of effects were shown is because your energies are very high. The excitation energies are right.
I see. Right.
Very high above KT.
Yeah.
So, and of course this is unique to light absorption systems because everything else in biology runs at about
Much slower,
say,
yeah.
half an electron volt.
Yeah. Yeah.
So I'm kind of interested in how this scales down,
Right.
down to half an electron volt from 2. Do you guys, can you generalize?
So I should say that energy scale you're talking about, the optical energy scale, right, is not relevant here.
Okay.
Because it is really in this it's really in the thousands of wave numbers what we're talking about, the disorder. Because it's transport in this in the excited manifold, right? It's really, I mean, the converse question of how it would scale with very large energy differences, I don't think it would scale well. I mean, you would get localized very quickly because at least to my knowledge, there aren't dephasing mechanisms that could bridge this huge energy gap.
Okay, so
I think that's the.
Let's make a thought experiment. Suppose that your photosynthetic pigments with mid-infrared absorbers.
Yeah, yeah.
So you arrange your
Yeah.
energies, your orbitals, energy levels to absorb at, let's say, half an electron volt.
Right.
What would it look like? I mean, you can actually do that in simulation, I assume. What would the entanglement look like if everything took place at the energies roughly of ATP hydrolysis, let's say?
Yeah. We haven't done simulations, so I don't know what it looks like, but it's an interesting question. I mean, I think the thing you have to be concerned about is actually relaxation mechanisms in that case, because the energy you have to relax is much smaller, right? So one of the things that photosynthesis has working for it, you get this initial kick of energy, you go up a huge amount, and then you just play around in this like large energy scale, right? Whereas if you push down where that excitation manifold is all the way here, you have a larger chance of losing the energy that you initially got. Yeah, so then you would, I think you would lose all quantum. Well, maybe I shouldn't be so general because I haven't done the simulations, but I think.
You don't actually believe it.
Well, I believe it about these systems, but I don't believe about an artificial or an infrared absorbing system, I guess. Yeah. Yeah.
I don't know if you're familiar with this. This is a very old story.
Mm-hmm.
Going back to the 1920s, the work of chemists in Germany and England
Mm-hmm.
under the name Scheib aggregate and J aggregate.
Yeah. Yeah.
Because
Yeah,
their last
decent.
foray into engineering of these systems reminds me of what they did in terms of engineering, light harvesting, artificial dyes, light clotted thin film.
Right. Right.
So perhaps you can get inspiration from that. Because
Yeah.
There was also a case where actual temperature promoted the efficiency of light harvesting.
Right. Yeah.
As opposed to what you expect from quantum mechanics, and the equation was quantum mechanical from collective states.
Right. Yeah, so we are very much inspired by that work, and so the artificial systems we think of as actually as J aggregates that are encased within a protein. So the protein degree of freedom is an additional degree of freedom from these other older aggregates that were engineered. Yep.
The tobacco mosaic virus is structurally similar to a microtubule.
Okay.
In fact, so two questions. You considered using that microtubule instead of a tobacco mosaic virus? And B, what does that tell us, or what could it tell us about how microtubules utilize their own geometry, periodic lattice geometry, not with chromophores, but with aromatic amino acids?
Yeah. Okay. So first, we haven't thought about using microtubules because there are certain advantages to tobacco mosaic virus because we know the synthesis pathways, we know exactly how it aggregates and so on, or how it self-assembles. I'm not sure. So I'm not a synthesis expert, so I don't know if the same thing is known for microtubules. The other thing is that this is not a natural thing to make these viruses do, right? They don't actually have chromophores in them. What goes up that helical chain is actually the RNA. So what you've done is you've stripped away the RNA where normally the RNA would bind and actually bound these chromophores. So the question about what in nature they wouldn't have chromophores attached to them is what I'm saying. And also we're more interested in the quantum properties of the chromophores themselves rather than the protein, the tobacco mosaic virus protein. The protein is really the environment for the chromophores, right? So we're interested in not necessarily engineering the quantum properties of the protein, which is a very difficult task because it's just a large structure, but rather the quantum properties of the chromophores. Does that answer the question?
Well, right, but you can also use the microtubule as a scaffolding and attach chromophores by microtubule-associated proteins very selectively, actually, probably
Okay.
more elegantly than you do with TMP.
Okay, so my expertise is in synthesis. I mean, this is the expertise that the lab that we work with has, but I'd be happy to hear about that. Yeah.
I think what we should do, we should now, first we should thank Mohan for the excellent